Once again, welcome back everyone to your favorite and my favorite biopoint stream, the world of bio. So once again, we are back with yet another session from molecular basis of inheritance, lecture 3, and the topics that are going to be covered in this session are transcription, genetic code, and types of RNA. So this is once again back with you from the team BioPoint as a part of Crash Course 2021. So hope you all are perfectly ready for this session. So once again, a very warm welcome everyone to this session. So do not forget to hit the like button, do share it with your friends, and do subscribe BioPoint Stream the World of Bio. So let's start with today's session that's on transcription. And as you all know, we have entered last class with the topic of with the different experiments conducted by different scientists, and today we are going to start with transcription. So the basic definition of transcription, or the definition of transcription, is that it is the process of copying information from DNA to mRNA. From DNA to mRNA. As you can see in this directly in this slide, it is messenger RNA, which is the mRNA. Is that clear? Fine. So the process of copying genetic information from one strand of the DNA into RNA is known as transcription. Okay. Okay. So then what is this genetic information? It is a particular method of arrangement of base pairs with genes. So what is this genetic information? It is the particular method of arrangement of base pairs with the gene, within the gene. Okay, inside the gene, the particular arrangement of these base pairs is known as what? Genetic information. So in between two apart genes, there is no information in that area, and there a particular base pair simply repeats. Okay, in between two gene, there is no information. That is, there is no arrangement. There is no base pair in that region, and at that region, what is there? There is a particular base pair that simply keeps on repeating, and such a simply repeated base pair, or they form a sequence known as repetitive sequence. repetitive sequence Repetitive sequence. In most of the DNA, major part will be the repetitive sequence, and if it is very large, if the repetitive sequence is very large, it is called VNTR. VNTR. Variable number tandem repeats. Variable number tandem repeats. This is known as VNTR, variable number tandem repeats, or satellite DNA. Or satellite DNA. Is that clear till now? Is that clear till now? Okay, fine. Fine. So here, adenine forms base pair with uracil instead of thymine. As you all know, in case of RNA, thymine is replaced by uracil. Is that okay? Fine. The DNA-dependent RNA polymerase catalyzes polymerization only in 5-3- direction. The direction of transcription, the polarity of transcription is 5-3- direction, and the 3-5- that is the opposite strand. 3-5- strand act as the template strand, and the other 5-3- that is the direction, that what The strand in the direction of polarity stands as the or act as the coding strand. Is that clear? Is that clear? Give me thumbs up on the screen if it is clear for you all. Okay, fine. Both the strands are not copied during transcription. So why both the strands are not copied during transcription? It is because only one strand is taken and the other just forms another new strand. One strand is taken and the other forms another new strand. So the code for proteins is different in both these strands, and if both these strands are taken in one RNA, what happens? That complicates the process of translation. What is translation? That is the next process after transcription. So if two RNA molecules are produced simultaneously, and that would be complementary to each other, and that forms a double-stranded RNA and prevents translation. Okay, we said that the code for proteins is different in both strands, and if you take both strands simultaneously, two types of RNA molecules will be produced, and that may be complementary to each other, and that prevents the process of translation to occur. Is that clear? Is that clear? Okay, fine. Transcription unit. Transcription unit. So, transcription unit is the segment of DNA between sites of initiation and termination of transcription. So what is a transcription unit? A transcription unit is a combination of P site, that is the promoter site, structural gene, and T site or the terminator site. The combination of P site, structural gene, and T site together forms transcription unit. And what is this promoter site? It is the binding site for RNA polymerase. It is the region where the RNA polymerase starts to bind with the DNA. Okay, and it is located towards 5' and upstream. Located towards 5' and upstream, like this. Okay, and the what terminator region is located towards 3' and downstream. 3' and downstream. And the region between this P site and T site where transcription takes place. Transcription takes place in the region between promoter and terminator, and that is known as structural gene. And that is known as structural gene. And this terminator is the site where transcription stops. Transcription stops at the region of termination. Okay, is that clear? Now it's time to remind you to join our Telegram group where we have the daily session updates, regular discussion, daily polls regarding the questions of NEET, and where you can interact with me. And other feature of our group is that we have handwritten test in our group where you have to self-evaluate yourself and send us the scores via the IDs that are provided to you along with the question paper. So guys, the direct link to join is given in the description box, and it is t.me/neat_biopoint. So hope if anyone new is watching this session will join the group right now. Okay everyone, so let's move on with our session. We have transcription unit and gene. The gene, what is a gene? Gene is the structural and functional unit of inheritance. It is the functional unit of inheritance, and it is the DNA sequence coding for an RNA. So genes code for RNA. What all types of RNA? mRNA, rRNA, and tRNA. mRNA, rRNA, and tRNA are the different types of RNA, and genes are the functional unit that codes DNA sequence for an RNA. Then what is the cistron? What is cistron? Next question is, what is cistron? It is the part of a DNA that carries information about one polypeptide chain. Okay, so a segment of a DNA coding for a polypeptide chain during protein synthesis is known as cistron. It is the largest element of a gene. Here it is heavy raining. It is raining heavily. So, and thunders are there along with that. So I don't know how much far will this session be continued. So let's do go on with that. If there is any issue with the audio, kindly use the headset or the earphones. Okay, guys. So fine. Structural gene in a transcription unit is of two types, that is monocystronic gene and polycystronic gene. Monocystronic gene and polycystronic gene. Okay, so the monocystronic structural genes or the split genes are present in eukaryotes. They carry information about one polypeptide chain. So the monocystronic structural gene carries information about one polypeptide chain. Polycystronic structural genes carry information about many PPC polypeptide chains. Okay, and these polycystronic structural genes are seen in prokaryotes. Okay, fine. Exons appear in processed mRNA. Introns do not appear in processed mRNA. So what is this exon and intron? Intron is the information absent area in an mRNA, in an RNA or an mRNA. In a what mRNA, if the information absent area is present, that is known as intron, and the information carrying or expressing area is known as exon. Expressing area, exon. Okay, information absent about information absent area, that is intron. Is that clear? Is that clear? Okay, fine. Transcription in prokaryotes. In bacteria, that is prokaryotes, synthesis of all types of RNA are catalyzed by a single RNA polymerase. So they are catalyzed by a single RNA polymerase, and this transcription in prokaryotes is of three types, that is initiation, elongation and termination. In prokaryotes, both transcription and translation takes place in the same compartment, that is in the cytoplasm, that is within the cytoplasm where the nucleus is absent. And the key enzyme for the process of transcription in prokaryotes is RNA polymerase. RNA polymerase is the key enzyme. Is that clear? Is that clear? Fine. So how many slides are left? Okay. So here RNA polymerase enzyme binds at the promoter site of DNA. This causes the local unwinding of the DNA double helix. Here, an initiation factor, that is the sigma factor present in RNA polymerase, initiates the RNA synthesis. So how is this transcription occurring in what? Initiation occurring in what? mRNA. That is what we are going to discuss now. The RNA polymerase, that is the key enzyme, they attach on the P site. Okay, what happens there? They attach on the P site, that is the promoter region. Okay, then the factor called sigma factor activates it. Sigma factor starts to activate the transcription, and that process is known as initiation. Is that clear? Is that clear? And the enzyme starts to move downwards, then two strands unwind each other. That is the 3-5' strand is called the template strand, and the 5-3' strand, which is called the coding strand. This starts to unwind each other. And moving on to the process elongation. RNA chain is synthesized in 5-3' direction, and in this process, activated ribonucleoside triphosphates, ATP, GTP, UTP, and CTP are added. And this is complementary to the base sequence in the DNA template. So what happens is that the RNA synthesis occurs complementary to the template strand, and this RNA synthesis occurs in 5' 3' direction, and that is known as elongation, where the newly synthesized RNA is same as that of coding strand except for uracil. So except in case of uracil, all others the coding strand is same as that of this newly synthesized RNA. Okay. Then next process is termination. That is the RNA polymerase reaches the terminator site. That terminator site, that is the T site, and RHO factor stop it. RHO factor stops this raw factor. Raw factor stops this process of transcription, and thus what happens is that the process of transcription comes out to an end in prokaryotes. RNA comes out, and enzyme separates, and DNA starts to recoil back. Is that clear? Is that clear? In bacteria, transcription and translation can be coupled. Translation begins before mRNA is fully transcribed because mRNA requires no processing to become active. Transcription and translation takes place in same compartment. That is no separation of cytosol and nucleus. There is no separation between the cytosol and nucleus. That is the both, as I have said you, transcription and translation takes place in the same compartment. Give me hearts on the screen if it is perfectly clear for you, if it is perfectly fine for you all. So in eukaryotes, transcription in case of eukaryotes is our next topic. Okay, guys. So in eukaryotes, there are two additional complexities. That is, there are three types of RNA polymerases. The primary transcripts, that is the hnRNA, which contains the exons and the introns, and are non-functional. Hence, the introns must be removed. Okay. Three RNA polymerases in eukaryotes, that is the RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. So the procedure of transcription is similar to that of prokaryotes, but after transcription, mRNA carries information. Just like split genes, mRNA carries information after transcription. Okay. And the newly synthesized RNA, that is the hnRNA, undergo three processes, that is splicing, capping, and tailing. Before moving on to that, let's discuss these three types of RNA polymerases. Oh my god. The RNA polymerase 1, that is transcribes rRNAs 28S, 18S, and 5.8S. RNA polymerase 2 transcribes the hnRNA, that is the heterogeneous nuclear RNA, and it is the precursor of mRNA. So the precursor of mRNA is hnRNA, heterogeneous nuclear RNA. Okay, fine. RNA polymerase 3 transcribes tRNA or 5S rRNA and snRNA, that is a small nuclear RNA. This is the three types of RNA polymerases. Processing of primary transcript, that is hnRNA. That involves three processes, that is splicing, capping, and tailing. Splicing from hnRNA, that is introns are removed by spliceosome, that is the enzyme, and the exons are spliced together. So removal of these introns and joining the exons together using splicing enzymes, that is the spliceosome, is known as splicing, and the length of RNA decreases in case of splicing. Next is capping. Here a nucleotide methyl guanosine triphosphate cap is added to the 5' end of hnRNA. That is, addition of a 7-methyl GTP, 7-methyl GTP, 7-methyl guanosine triphosphate. Such a cap is added at the 5' end of the hnRNA, and such a process known as capping. And what is tailing? That is polyadenylation. Polyadenylation. That is at the 3' end, 200 or to 300 adenine bases are added. That is known as the poly-A tailing or polyadenylation. Poly-A tailing. Now it is the fully processed hnRNA called mRNA. So that's all for the process of transcription. Now we are moving on to genetic code. Till then, is that clear for you? Till now, is that clear for you? Okay, everyone. So hope it's perfectly clear for you. And now we have the next topic of genetic code. So who all decoded this genetic code? Which all scientists decoded this genetic codes? We want to know that, right? Anyway, we are going to learn this topic, so we want to know who all decoded this genetic code. Okay, that is first one is by a scientist called Marshall Nierenberg. Marshall Nierenberg. Second by a scientist called Mathai. So here A is there, then EI. By a scientist called Mathai. Third, by another scientist called Kornberg. 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 Fourth, by another scientist called George Gamow. George Gamow. Fifth, by another scientist called Severo Okawa. Severo Okawa. Okay, fine. The last by H.G. Corana. H.B. Corana. H.G. Corana. H.G. Corana. So all these scientists together decoded genetic code. Marshall Nierenberg, Mathai, Kornberg, George Gemma, Severo Okawa, H.G. Corana. All these scientists together what decoded this genetic code. So what is this genetic code? It is a sequence of nucleotide, that is nitrogenous bases in mRNA that contains information for protein synthesis or translation. That contains information for translation. So the sequence of 3 bases determining a single amino acid is called a codon. So what is a codon? It's a triplet, and this triplet is formed by Nierenberg and Mathai. Nierenberg and Mathai. So these two scientists constituted this, discovered this codon, that is triplet constituting a single amino acid, bases constituting a single amino acid. So all the different types of amino acids that we have discussed in, you have learned in biomolecules, right? So George Gamow suggested that coding 20 amino acids, the code should be made up of 3 nucleotides, and thus there are 64 codons. So how many codons are there? There are a total of 64 codons. There are a total of 64 codons. Hargobind Khorana developed the chemical method in synthesizing RNA molecules with defined combination of bases, that is homopolymers and copolymers. Then Marshall Nirenberg developed a cell-free system for protein synthesis. Cell-free system for protein synthesis was developed by Marshall Nirenberg, and Severo Okava enzyme, that is the polynucleotide phosphorylase, is used to polymerize RNA with defined sequences in template independent manner. Okay, so these are the what discoveries of all the scientists that we have discussed, and the 20 types of amino acid: alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, Rheonine, tryptophan, tyrosine, valine. All these things we have to learn in biomolecules. That chapter 2 we will be covered before the NEET 2021. Before the NEET 2021. Okay, everyone? Fine. So this is the codon for various amino acid. That is the 64 codons and amino acids. 64 codons and amino acids. And now what I have to remind you is that the updated playlist of the Crash Course 2021 is given there in the description box. Do not forget to visit that and utilize it maximum for your educational works. And it's from the team BioPoint, Stream the World of Bio. So do not forget to utilize it maximum. And it's prepared for you only in order to have maximum works for you in order to clear your doubts, and without any fail, without wasting any time, directly you can visit the playlist for clearing any of the doubts in the chapters that we have taken for the Crash Course 2021 in one shot. Okay, everyone? Then, how many slides left? Actually, heavy rain and thunder is there, and it's hard for me to take it. 1437. Okay, let me move fast. Codon is a triplet, that is a three-letter code. 61 codons code for amino acid and 3 codons UAA, UAG, and UGA. Do not code for any amino acid. Okay, and they act as stop codons or termination codons or nonsense codon. Guys, silent features from the silent features of genetic code. Let me take from next class. Do you know because of why? If I am taking it today because of this heavy rain, I cannot focus in my class. And what happens is that the topic will not be moving smoothly. That's why even the process of transcription, I don't know that I have moved a bit fast, and it was not at all clear for you. I don't know that. Okay, and why I just again moved on with that is again this NCRT highlight is coming on this chapter after the entire session. That is, this is transcription. After that, two more sessions are left. That is one translation, and the other one is about genome project. That too will move very fast. The last class will be minute of 20 minutes or 30 minutes. Only that's enough for that class. So hope everything will be moving fastly, and thank you all. Thanks for your patient listening. It's me signing out from your favorite and my favorite biopoints in the world of bio. So once again reminding you, do hit the like button, do share it with your friends, and do subscribe to the channel Biopoints in the world of bio. Bye bye.